So our findings for this study. With the field study, pretty simple, pretty straightforward. It was a big undertaking, a lot of samples, a lot of logistics, either traveling or getting samples around, but the results are pretty simple. Basically, going from Arizona being the hottest place we sampled down to North Dakota being the coldest. Relatively clean trend. The warmer it was, the more often we found Legionella, and to some extent, the more there were there. Not significant differences, but the numbers for the three warmest climates were much higher than anywhere else we saw the Legionella. And when we repeated our sampling from summer to winter, those were summer results, by the way. In winter, we figured temperature is going to be a factor. More people, they're going to be more likely to use higher concentrations of methanol in their washer fluid during the winter. We only found Legionella in our hottest site in Arizona. So in winter, everywhere else got freezing temperatures. We didn't find any culturable Legionella. So based on those results, we were thinking, hey, our hypothesis is kind of filling out. Like, it makes sense. Hot temperature, more methanol. Hot temperature is good for Legionella, less methanol is good for them, so warmer climates are better. Now, the results get a little tricky for our bench experiment because almost the exact opposite happened. So without going too much into detail, it's a lot of sort of data to kind of go through, but giving the big results. Basically, when we incubated them in the same washer fluid from our Michigan, one of our cold weather sites, and our Arizona washer fluid from our Arizona site, they survived better in the Michigan washer fluid than the Arizona washer fluid, even though the Michigan washer fluid had 35% methanol by volume. So a very high methanol concentration. So when we were first starting this study, we saw very high die-offs in any washer fluids with methanol, which we figure makes sense. It's an alcohol, bacteria, at least Legionella. Many bacteria have a hard time handling being in a solution with a lot of alcohol. It does a lot of harmful things to their cells, right? So we figured, okay, if they're dying, it makes sense. Except in our Michigan washer fluid, they weren't dying, and eventually everyone else died off, even our positive control. In our just plain tap water we had, they started eventually dying off, which we were thinking, okay, something funny is going on. Repeated it, same deal. So that was with culturing. When we looked a little closer, basically when we performed microscopy on these guys, we could tell if they were alive, but sort of in a hibernation state, in a dormant state, where they didn't want to grow. And that is what was happening. So across the board, except for the highest concentrations of methanol, very little death was occurring in these Legionella. They were just becoming stressed out to the point where they didn't want to grow, but they weren't dying in any sort of huge way. So from our Arizona samples, that looked like they died off very quickly, almost entirely there. In our Michigan samples, they were alive, they weren't dying, they just were healthier to the point where they were able to grow. Why that happened, we don't know. One other interesting fact, interesting piece of data that came out of the study, and sort of a big point for some of our follow-up work we want to look at, was the methanol concentration in the sample that had the best survival and that Michigan washer fluid. It decreased. We're not 100% positive, but it went from about 35 down to about 15%. So noticeable, measurable, potentially biologically relevant decrease in methanol concentration. We have no idea why. It was basically washer fluid. It had been sitting on our shelf at room temperature for months before we used it. It didn't decrease hardly any in concentration. We incubated it with just Legionella, same temperature, same conditions, and it decreased. So why that happened is it was almost a, I don't know, side note. We weren't really predicting anything to happen with methanol. We just wanted to make sure the concentrations were as they were on the label, but it decreased. So that's maybe an interesting result that came out of it, sort of, I guess, a lucky result that came out of it.